Okay, welcome to next session of bioinformatics. I am Dr. Y.V. Rajesh. So we'll learn about pairwise sequence alignment methods, and we'll learn more detail about dot plot approach. So previous sessions we learned about definitions of sequence alignments. We learned about global alignment and local alignment. So we'll see the methodological part of sequence alignment, how it is done. So if you have two sequences, sequence 1 and sequence 2, so there are four broad approaches are there: by hand alignment, dot plot method, heuristic methods, and dynamic programming approach. Four different kinds of approaches you can do here. So hand alignment, basically what you would do is you take two sequences, note down on pen and paper, and try to match the sequences, the matching positions in two sequences. So that will become hand alignment. Dot plot, it is a graphical visualization method. Heuristic method, algorithmic approach, but the approach is heuristic, and dynamic correct approach through proper programming approach. So we'll see about hand alignment followed by dot plot methods in detail here. So by hand method, what we do is we take two sequences. Let us assume that I have two sequences, TC-ATG, CA-TTG, two sequences. So these two sequences can be aligned in four different ways when you are taking something called as gap. So there is something called as a gap. Gap is nothing but keeping a dash in between two characters of two sequences here. So gaps can be, it can be located at the beginning or at the end or in the middle of the sequence here. So anywhere you can have the gap here. So computationally you insert gap to match the maximum number of matches here, and biologically it has very importance here. So we know that DNA undergo mutations. So mutations broadly classify, if you want to classify mutations here, so you can have the mutations can be point mutations or frame shift mutations here. Point mutation or frame shift mutation here. So what is a point mutation? So a character is changing to another character. Simply let us assume that I have a DNA sequence. So in one generation my DNA sequence is like this: AT, G, CAT. So when it is passing to next generation, it is becoming ATG and GAT. At fourth position you have C in one sequence and G in another sequence. So this will become a point mutation here. Okay. So now take another case here. So our sequence ATG CAT, and we'll take some more extension. You have another set of characters TAG. Now in next generation it is becoming ATG, then CTT and AG. So here what happened here is you have three codons in first sequence, and in second one you have first codon is matching, and but for second codon this mismatches happened because of deletion of A when it is going to next generation here. That means A deletion is leading to the frame shift in the codon positioning here, frame shift in the codon here. So here entirely the complete positions or complete frame will be shifted here. So when you keep an insertion here, so what we are making is assuming that there might have frame shift mutation occurred in the DNA sequence. So because of that one, the matches is not happening properly. So to circumvent or to take out of the frame shifting here, we insert gaps here. So now you see, first thing is without inserting gaps, I have TCA-TG and CA-TTG. So here I can see, so here actually the positions are matched, slightly changed here. TG in first sequence and TG in second sequence, both are matching here. So two matches and zero gaps. and next you take same sequences, but here a gap has been incorporated in the middle of the first sequence, leading to the gap at the beginning of second sequence. So TCA dash TG and dot or dash CA TTG. So because of this one, CA matching, TG matching. So 4 matches and one insertion or one gap here. So this is second one, and third one here. So I have kept 2 end gaps in 2 sequences. Then CAT in first sequence and CAT in second sequence are matching here. Now fourth approach, I'm inserting a gap just before the last character. So TCA T dash G. So and second one dot CA TTG. So because of this one, 4 matches are there. So now out of all 4 approaches or 4 different kinds of alignments here, I can say that the second and fourth one have more number of matches here. So like this we try to do by hand approach, and by hand method is actually not applicable for real life sequences, because real life sequences are very long. So if you take a human chromosome, the human chromosome one is actually 240 million characters are there here, and if you take simple proteins also, on average 100 amino acids are there. So the comparisons is not possible or not trivial using by hand method. That's why we need the use of computers here. So that's where the bioinformatics play the role here. So the definition of bioinformatics is using computer science for managing biological data, simplest definition here. So the second method, which is a visualization method or graphical method of pairwise sequence alignment here. So the pairwise sequence alignment, the dot plot alignment, it gives a visual overview of all possible alignments. So it